Susanti. Without any further ado, let's start the expert talk, the session 2, by listening to the welcoming and opening remarks from Pak Kapus. PRAA, please welcome Pak Asep Hidayat, PhD. The time is yours.
Yeah, thank you, Mr. Bugi, from my time. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. In here, I would like also to express our appreciate to Dr. Mika Horicek. This is right, sir? Yeah, I'm sorry. Now he is a visiting researcher in the BLIN with the Dr. Juliana as the host from visiting research program. And also I would like to thank from him for your time. Then he will deliver your expertise, I think, in the expert talk in the second session. And then I would like also thank you to jamming session team. Also, I would like to say thank you to the Dr. Ruliana and then Mr. Bugi, who is have expert talk today. And then I hope this expert talk will deliver theme related to the application of the stable isotope technique for different field of the research. And then I also hope by this event it will have a chance to share knowledge or collaboration to make a strengthen of our research output, as well as making deep research in the field of the ecology and ethnobiology. And I hope also in this event all participants in the expert talk today will make good discussion with Dr. Mika, I think. And then I hope using the discussion we can have some encourage or improvement to our research in the future. And maybe in the end, I would like to also thank you again to Mr. Mika and another participant for your coming in the expert talks in the second session. And then I hope using this expert talk, we will have your information or your expertise, then using one tool to identify the originality of some species, maybe Mr. Mikan later will more give explanation more detail. I think this all my special speak maybe and then I will give again to the Mr. Bugi as the MC. Thank you, Mr. Bugi.
Thank you very much, Pak Kapus, Pak Asep Hidayat, PhD, the head of the Center for Ecology and Ethnobiology, for a welcome speech and has officially opened the expert talk session second edition. Before we continue, we have already 74 participants here in the Zoom. We are going to have a photo session that will be handled by Mbak JG. Please, Mbak JG.
All right. Maybe I need an assist for Mbak Lutfi.
Okay, Mbak, I will start. There are 3 slides in my monitor. I will start with first slide. Please keep your smile and open your camera. Okay, 1, 2, 3. Second slide. 1, 2, 3. And last slide. 1, 2, 3. Okay, Mr. Bugi.
Thank you, Mbak Lutfi. I think we have already give a very best smile and photo for Mbak Lutfi to be documented. Now we are coming to the essence of the Expert Talk second edition, to the presentation of Dr. Misha Horacek that is entitled Application of Stable Isotope Technique for Different Fields of Research. Dr. Misha is from Austrian Agency of Health and Food Safety, Vienna University. And this session will be led by Dr. Juliana Susanti, a senior researcher from Research Center for Ecology and Ethnobiology. But before that, allow me to give a little bit of nuance for this presentation. I will read a little pantun or poem. In every leaf, God's grace we see. Ecology's song in every tree. Ethnobiology whispers his plant, a sacred bond between earth and man. And now, Buruli, the time is yours. Thank you.
Thank you very much, Pak Bugi. Good afternoon, everybody. All participants, thank you very much for joining us today. I'm pleased to introduce our distinguished speaker, Dr. Misha Horacek, who will be leading today's webinar. Dr. Horacek is a highly respected expert in stable isotope geochemistry from Austria, with a wealth of experience in applying techniques for provenance studies, particularly in tracing the origin of natural resources such as wood, food, or animal products. Dr. Horacek currently works in Austria Health and Food Agency, or AGES, and also affiliated with Vienna University in Austria. He has been at the forefront of research in this field, and his work has significantly advanced our understanding on how stable isotope can be used to verify the origin of wood material or traded wood materials. His expertise has been instrumental in shaping our current research initiative, and we are honored to have him share his insight with us today. Without further ado, please join me in welcoming Dr. Misha Horacek. You have all the time, Misha.
Thank you, Juliana, for this nice introduction. And also for me, without further ado, I will come to start to my talk. Just for your information, my talk is planned to consist of 3 parts, presenting 3 different topics of my research. I will see, with respect to the time, whether I will have time to present all 3 of them. If I will be running short of time, then I will stop after the second presentation. But now I will come to the first one. So let me see. Can you see it? Okay. So the topic of my first presentation is the control of geographic origin of Tyrolean milk by stable isotope analysis. What's the aim of the project? Customers are willing to pay more for goods, especially for food, coming from certain regions or provinces. Tools need to be installed to control the origin of food products to ensure the correct declaration of origin, because it has already occurred cases where food products have been sold incorrectly, labeled, is coming from a certain area. Why do we want, or we have to do this? First, to protect the goods coming from these certain areas, and most of all, to protect customers from being deceived, and to retain and increase the customers' confidence that the declared origin of a food product that they are buying is controlled, and it's actually also coming from this area. For the control of this claim, we apply stable isotope analysis because it has been proven that just controlling paperwork is insufficient, as paperwork can even easily switched, faked, or let's say exchanged, and so the control of the product itself is required. Just a short introduction about stable isotopes. Most elements exist in forms of different weight, which means a different variable number of neutrons in the atomic core, together with a certain number of protons, and these are the isotopes. The abundancy of the heavy isotopes is usually much smaller than the abundancy of light isotopes, and the ratio of the heavy with respect to the light is expressed as the isotope ratio. And most, or let's say, the majority of the elements have stable isotopes, which means they don't decay with time, and unless there is any change in the product itself, the stable isotopic composition of the product remains stable and does not change. With the laboratory I'm working in and I'm heading, I can analyze the isotopes of 5 elements. These 5 elements are hydrogen, carbon, nitrogen, oxygen, and sulfur. And here in this table, on this slide, you see the light and heavy isotopes of these elements that we are investigating. With respect to oxygen, it's only O16 or O18 versus O16. Why do we analyze stable isotopes? Because stable isotopes are fractionated with certain environmental processes. And one of these processes is the water cycle. And this water cycle influences the hydrogen and oxygen, the elements water is consisting of. Whenever water is evaporating from a reservoir, let's say a sea, preferentially the light isotopes go into the gaseous phase and form water vapor, is shown here in this sketch, whereas preferentially the heavy isotopes remain in the liquid phase. And if the water vapor forms a cloud and starts raining out the water, preferentially the heavy isotopes that were transferred into this cloud first go into the liquid phase again, whereas the light isotopes are rather remaining in the water vapor, which means that if this cloud is transported over a continent or over a distance, it gets successively isotopically depleted. This you can see here on this map sketch on the right side, where you can see heavy values, which are these orange-yellow colors, are getting towards lighter colors towards the east, and also from south to north, because we have 2 effects. One effect is the continental effect, which means if a cloud is traveling over a continent and is now and then raining out, it gets more and more isotopically depleted due to the continental effect. And the second is the temperature effect. When water evaporates at high temperature, also many heavy isotopes get in the gaseous phase, versus evaporation at very low temperature, almost no heavy isotopes transport into the gaseous phase, which can be seen here on this sketch as well. You see the gradient from south to north. Reasons for isotopic functionation for other elements, for example, as carbon, have different, let's say, processes. And here one of the important and relevant processes is photosynthesis. As plants have different types of photosynthesis, this is expressed also in the carbon isotope composition. For example, C3 plants, they perform photosynthesis in the Kelvin cycle, and as a result have a carbon isotope composition of around -20 to -30, whereas C4 plants, they have photosynthesis following the Kelvin cycle plus the Hedge-Slack pathway. They have a carbon isotope composition around -10 to -15, and in between there is the CAM plants, but they, I would say, usually of lesser relevance, and I don't want to go into detail with respect to that. Besides photosynthesis, there's also a second parameter or process influencing the carbon isotope composition. It's the water availability, or better said, the absence of water. When there is drought stress, plants increasingly integrate also heavy isotopes in their plant matter in the photosynthetic path than plants that do not experience drought stress and have sufficient water available. Here is just a short sketch showing the ranges, the isotopic ranges of different let's say commodities and materials. Of relevance is here, what I just want to show is in the upper right parts is the C3 plants and the C4 plants. You can see that they have a quite range of variety, but are almost non-overlapping. The rest is now not of such a high relevance for this talk. For the isotopic fractionation of N, of nitrogen, we have again different processes, and here it is the process of bacteria which transform nitrogen and can fixate nitrogen, and also the abiotic fixation of nitrogen in the soil. For plants, so to say, they incorporate nitrogen without a big fractionation, what is available in the soil, with the exception of the leguminose plants, which are nitrogen fixator that can access nitrogen directly out of the atmosphere and therefore have a different nitrogen isotopic composition than non-leguminose plants. Animals are more enriched in nitrogen than the food they feed on, because there is some fractionation in the feeding, or let's say in the animal metabolism. And this can also be used with respect to food webs, as whenever one animal is consumed by another, and you are moving upwards in the food chain, the nitrogen isotope composition is increasing. Means that the top predators also have the highest nitrogen isotope value with respect to the animals at lower positions in the food chain. Sulfur isotopes are almost non-fractionated in biogenic processes, or at least in mice biogenic processes. There are exceptions, of course, but with the processes taken here into account, there is almost no fractionation, but they are fractionated by geogenic, let's say, processes, and therefore they are called a geogenic parameter, because different geology can have an influence on the sulfur isotopic composition, and thus different geological, let's say, realms are also the reason for different sulfur isotopic composition of the plants living on these realms, in these realms, and the animals feeding there. Additional sources of sulfur are also volcanic emissions and sea spray. And of course, also human activity can be a reason of sulfur introduction in the environment. The influences of the isotope ratio in the plant can have many parameters. It might be the species, as I said, photosynthesis, and also the rate. But there are several, I'd say, parameters, and the list is non-complete one. And the second one is the environmental. And the environmental process influencing the plant. These are the relevant ones right now, I would say, because it's the environment that defines the the isotopic composition of a plant and or then the animal feeding on these plants. And so we are mainly interested in the environmental conditions specifically for certain places and regions. As I said, for the animals, it's rather easy because the animals, their isotopic composition is determined by what they feed on and the water they are taking, they are living on. So you can speak more or less, you are what you eat, plus a few per meal, as it said, because the metabolism, let's say, is the reason for some slight, let's say, fractionation for several of these elements. So now come to the study. We studied Tyrolean milk because Tyrol is a mountainous area which is claimed of producing milk of very high quality, and we want to protect this product. And over a period of 1 year, 15 dairies have been sampled monthly throughout the entire Tyrol state, and the milk was processed and analyzed for hydrogen, oxygen, carbon, or nitrogen. And a few non-Tyrolean samples were also collected to check whether a differentiation between Tyrolean and non-Tyrolean origin is possible. Just a quick environmental setting. Tyrol is in Western Austria. The latter is situated in Central Europe. Tyrol is a mountainous region in the Eastern Alps. The dairy cows in Tyrol usually, or let's say very often, graze in summer on alpine meadows in the mountains and are kept in the stable in winter, feeding on hay, dry feed, whereas on the meadows, of course, they're eating the fresh grass. Here is the small sketch of Tyrol, and you have the large, let's say, part here. This is called Northern Tyrol, this one here. And here, this small patch here on the east is Eastern Tyrol, and here on the south is Southern Tyrol, which doesn't belong to Austria anymore, but it belongs to Italy. It's the northernmost part of Italy. And here is, in short, the dairies that have been sampled. And also here on the right side, you see the names of the other Austrian provinces where the non-Tyrolean milk samples have been collected from. And what you can see is these non-Tyrolean samples are from very nearby, which means that the environmental conditions should not be too different. And it's expected that if these samples can be differentiated from the Tyrolean milk, it is to be expected that samples coming from elsewhere, for example, from Italy, from Germany, from Holland or other areas, the milk from other origins should be even more easily to be differentiated from the Tyrolean samples. Here we have some the results for hydrogen and oxygen. You can see that this is a positive correlation, as to be expected, because both parameters are influenced in the same way. And we can see in short that we have a quite good differentiation between winter milk, where the cows are in the stable, and the summer milk, where when the cows are on the alpine, dominant on the alpine, feeding on the alpine meadow feed. You can see it approximately. Here is the border between these two, let's say, areas. And we can also see that the non-Tyrolean samples are here not, let's say, ideally to be separated, but it has become more easier, and the separation becomes, let's say, better by looking at the next graph. Let's see what we have here. Here is the oxygen versus carbon isotopic composition, and we can see here with this black line that on the left side of the line we're having the winter samples showing very low oxygen isotopic values, and on the right side we're having the summer samples showing elevated values. For the non-Tyrolean samples, we see that the winter samples are the open squares, and we can see that they're all on the other side of the line, so we can differentiate the non-Tyrolean winter milk from the Tyrolean winter milk, And for the summer samples, we see that we have here in the circle or in the ellipse the non-Tyrolean summer milk samples, which are, let's say, close by, but not within the cloud of the Tyrolean milk samples. So there also is let's say, the possibility of differentiation of non-Tyrolean milk from Tyrolean milk. What we also see here, as we have, let's say, shown the samples according to their sampling date, that we are having differences throughout the year. So we have variations throughout the year. We can see this here very nicely for the oxygen isotopic composition, where we have on the x-axis the months. Starting in March is number 1, until 12 is the February of the following year. And where we can see that we have the elevated values during the summer months, and the lower one during winter months, as said before. And we also see this, but to a lesser extent, for the carbon-13 values, and we have slightly lower values in summer than with respect to the winter. And this is due that in winter, when the animals are stable, that there is some feed of maize with C3 feed. What we also see is that we are having, let me see, we are here, one dairy is this brown circle. This is organic dairy, which is not allowed to feed maize to the cows. And for this reason, the milk from this dairy almost has the most depleted carbon isotope values. We also then did a comparison between northern Tyrolean and southern Tyrolean milk, which is the milk coming from the northernmost Italian province over the period of 1 year. Here again, we repeat the variation with the seasonality in both of the origins, in northern as well as southern Tyrol. But we see that there is a huge or a main overlap of these two regions. And the same is for carbon isotopes, even though we see there that the northern Tyrolean milk has slightly lower values than the southern Tyrolean milk. But again, it's a big overlap. And for nitrogen isotope, it's a complete overlap. This was no surprise, because the southern Tyrol area or region is of similar topography as the other Tyrolean countries, meaning that the environmental conditions are very similar, and thus also the product is very similar. But we see that as we have shown the differentiation between Tyrolean and non-Tyrolean milk, this applies as well in the same way to the southern Tyrolean milk. So I come to some quick conclusions that Tyrolean milk from northern and from southern Tyrol can be discriminated from non-Tyrolean milk. Among these two groups, there cannot be discrimination directly. Only if we identify individual dairies, it might be possible to differentiate the origin, but then you really have to look at the individual dairies, whether it's possible to differentiate. The next conclusion is, if we can differentiate milk, we can also differentiate milk products. It means all products where the milk or parts of the milk are included without addition of any other materials, such as yogurt, cream, sweet cheese, also cheese. What we also see is that to some extent, summer milk from cows in stables can be discriminated from cows grazing on meadows, because they have a different feed. And also organic milk might be discriminatable from conventional produced milk, but this is something rather tricky, because in theory, you can also feed the cows organic maize if it is allowed by the, let's say, by the definition of organic milk. And so you need to have certain preconditions to be able to differentiate between organic and conventional. So, as I said, stable isotope composition is also applicable to milk product, also for cheese. But with cheese being let's say, a product with longer shelf life, I have to take into account that it might become difficult to evaluate whether the cheese I'm investigating is produced from summer milk or winter milk. And then I have a larger overlap. And then it might become a bit more difficult to differentiate between the geographic origin. So we have to take this into account when investigating cheese or other products of milk with a longer shelf life. And as I've shown with respect to the organic farm, the use of maize as a feed component can be controlled by carbon isotope composition. It's in such cases where the use of feed of maize might not be allowed, and then stable isotopes can be a means to control this claim. So, and this is the end of my first talk, and I will switch now to the second one. Should there be any questions, I would like to ask you, please write them or remember them, and please ask them to me after the end of all my presentations. So just for a minute, I will.
There are already a bunch of questions for you, Misha, so do not worry.
Okay, no problem. I will try to answer all of them as best as I could, but I will now switch to my next talk. So the next talk is about something different. It's about, well, it's still the same. It's about the control of geographic origin, but now the commodity is wood. It's tropical timber. So it's the title is Stable Isotope Investigation of Triprochiton scalocylon, with the, let's say, the common name of Ayus. It's in a tree growing in Central Africa, and it was part of a large ITTO, the International Timber Trade Organization project. Its main occurrence ranges from Sierra Leone in Western Africa to the Democratic Republic of Congo in very center of Africa, the country in earlier times called Zaire. Currently, it's not certified by the FSC, but it's endangered due to overexploitation, as it is a species which is not, let's say, grown in plantations, but is entirely harvested in primary woods. This project was a large international project where 3 stable isotope laboratories and 2 genetic laboratories were involved, and authentic samples were collected from sampling teams in Ivory Coast, Ghana, Cameroon, Congo, Democratic Republic of Congo, Gabon, and Kenya. No plantation timber is included. Why? Because plantations is something what is especially difficult for, let's say, teams working with genetics. Altogether, more than 4,000 samples have been collected and analyzed. All of these samples have been analyzed for genetics, and almost 1,000 samples were analyzed for stable isotopes. So you see there is a bit of imbalance between the analysis for genetics and isotopes. And then at the end of the project, separate samples have been taken by 2 institutions to organize blind tests. For Ayus, which our lab, the species our lab was working on, around 160 samples were analyzed for isotopic patterns. They were analyzed for hydrogen, carbon, nitrogen, oxygen isotopes, and a subset of these samples were also analyzed for sulfur and strontium isotopes. And as said before, 20 blind test samples were analyzed. As again, the differentiation of by stable isotopes is possible due to differences in the ambient environmental conditions of the respective places. So this can be climate, soil, geology, water availability, humidity, topography, distance from sea, altitude, and even more parameters, because there are many potential influences on the environment. And hopefully, so to speak, these environmental conditions make a locality unique in a way that it will be easy for us to differentiate it from another. So here are maps showing the localities where the samples have been collected. And you can see here one problem, that for the samples coming from Congo, you may see it here, all the samples are coming from one spot, which of course is a non-ideal situation, but it's something that we had to deal with because that were all the samples that we received from Congo. So we got further problems with the reference samples and blindness material because different kind of reference materials was provided. There was provided drill core material from drill cores taken perpendicular to the growth tree, to the growth direction of a trunk. There were tree shavings. It's what has been cut from the tree trunk or from log by a big knife. And tree bark material has also been provided. Further on, some of the samples came from juvenile wood samples, not from adult trees, which was a further problem. By definition, or as it was defined for the project, only drill core material should be used or was in agreement with the sampling protocol. Nevertheless, we had to take what we received. So as already said, which parameters have been investigated. And we can come here to the results. And we can see here, this is showing oxygen versus hydrogen values. And we can see that there is partially a good separation, but especially the differentiation between Congo and Democratic Republic of Congo. These are the orange versus the yellow sample dots. This might be due to different sampling materials provided. So the situation is a bit more difficult than we have hoped for. For carbon and nitrogen, this is something similar, because also here we see a quite good differentiation between, again, for the Republic of Congo and Congo. And also here, it can be ruled out that this is based on the different sampling material. For sulfur and strontium isotopes, at least, the sample material does not play a role. So there is not to be expected that the material has an influence on these parameters. And what we can see here is that we have a quite good differentiation between Ghana and Côte d'Ivoire, the Ivory Coast, and those two from the other countries, such as Democratic Republic of Congo, Congo, and Cameroon. But if you look at the orange samples, you see that they are more or less all lying on one line with respect to the strontium isotope values. And this might be due to the fact that all of these samples come from one locality more or less in Congo. So this also might be a sampling effect. So afterwards, some blind tests have been carried out on these 20 samples. And it was mainly, so to speak, a competition between methods. Which method is better? Is it isotope method or the genetic method? And as a result, here is let's say the species that we have worked on. One has to take into account that the isotope group had a far lower number of samples or reference samples analyzed than the genetic group, but the outcome, the outcome was, let's say, not comparable, but it was that throughout the species investigated, the stable isotope group got a better result than the genetics group, which was for us, so to speak, of course, a positive result, because there still is a kind of competition between these two methods with respect to the provenance of wood. But of course, in the end, the main goal should be to combine both methods to get an even better result for the control of the geographic origin. This is something that still needs to be done, but and is on the way, or let's say one is working on this, but it is quite complicated because the type of data produced for both methods is very different, and so it's difficult to integrate these two types of data. I just briefly want to give you a few examples of other, let's say, studies on geographic origin of wood. Here is one study on larch wood, where we differentiate European larch from Siberian larch. And this can be done on the one hand, as been shown on the upper right figure, by using oxygen 18. And on the lower left, we can see it's also working for hydrogen isotopes. Both is working very well. And we have another example for what's called, for the Christmas tree, where the hemlock tree, where there also is the, let's say, the question whether this tree is coming from Austria or other origin, because let's say consumers often want to have a tree coming from their own country and not an imported, so to speak, foreign one. And there is also quite a good potential to differentiate these different origins. So I also come to some quick conclusions. We made a first step to control the declared origin of triplorhydron xeroxylon, the IUs, the tropical timber, because of course the study we carried out is not, let's say, sufficient to do a thorough investigation, but it is the first step to further continue working on this topic and to further analyze more samples and to let's say aim to let's say fill the results in a database to be able to then control on a routine base these wood species we have worked on. And the good results we are, of course, have been very happy about, that we have shown, despite having analyzed a lower number of reference samples than the genetics group, and especially the species we have worked on, the IUs one, which received the lowest amount of reference samples. But of course, as has been stressed by me right now, for routine control, further reference data are never necessary. Nevertheless, stable isotope analysis is already applied on routine base for origin control of wood and timber material, but it's something that is still in progress and is still growing and continues to grow. And the advantage of isotope method is that certain environmental conditions result in certain isotope ranges. As an example, I want to name here the geological parameters. If I have a certain geology, I can expect a certain range in the isotope values. And if I know this, then I can do some predictions of the isotope ratios I should expect for a certain area. Yeah, here I also, I'm at the end of the second talk, and I will just quickly now go to the third one. Let's see. This third topic is now again a slightly different topic. It's the investigation of stable isotope ratios of feathers from a waterfowl species, and it is specifically a specific species. It's a migratory species. It's the bar-headed geese, and we want to let's say we want to obtain information on the migration of these species, and especially of the individuals that we investigate. This study was partially funded by the FAO, and it was carried out within, let's say, one of the reasons we did this was as the avian influenza was a topic there, and one wanted to learn more about animal migration. The migration of birds has been a question to Maine since quite a long time, because Maine observed that birds appeared at certain periods of the year, and then they left again. And Maine asked what happened to them, where did they go to, do they return or don't they return? And this was a rather long debate, what is happening to these animals, these migratory animals. One first clue was obtained in Germany in 1822. A stork was shot having a spear through his body. So he was still alive when he flying with this spear in its body. So from the spear, it became clear that this specimen must have come from Africa, as this spear was only known from that region. So that was the first clue where migratory animals in Europe migrate to in winter. But of course, we want to have a different approach, not this is rather cruel one. Still, what we did is we investigated the feathers and investigated them for hydrogen, carbon, nitrogen, oxygen, and sulfur isotopes. As I said, animal tissue isotopes mirror the situation or the conditions of the ambient environment, plus again a few per mil. The samples that we have received and we investigated, they came from Mongolia. In Mongolia, the Temple Lake is an important, let's say, spot for this breeding of the bar-headed geese, and also for molting of the bar-headed geese. And so we obtained from 16 individuals which were caught there during the period when they were unable to fly due to molting. The 16 individuals have been caught, and 3 secondary feathers were removed directly from their body and were sent to us in the laboratory for analysis. And the aim was that we intended to produce, let's say, an isotope, let's say, range of the Temple Lake animals breeding and molting at Temple Lake, that we can identify them later on if animals are caught elsewhere. And then we should be able to identify them by that isotopic pattern. The bar-headed geese are migratory waterfowl, as already been said, overwintering in India and breeding and molting in Central Asia in summer. As said, Temple Lake is an important summer site for breeding and molting. I already told you about the hydrological cycle, so I will just skip this, because this we already talked about, that we have the, let's say, the fractionation in the water cycle. And we know that we're having in Siberia, here the example for Central Siberia, we have lakes and other water, let's say, water spots with very variable sulfur isotopic values. This is something which can be, on the one hand, interesting because we can differentiate these, but it can, of course, be a bit of a problem if we have a large variety of, let's say, results. When we look at our samples, we were quite surprised, because what we did see is, on these 2 diagrams, the x-axis is always the hydrogen isotope value, and we see that we have a large variety in ranges, in the range of the hydrogen isotopes, which can be interpreted as a large variety of the spots where these feathers have been built, which means low values, which are always on the left side. They must have been formed during molting at let's say a cool and continental area, whereas high values on the right side, they must have formed in very warm, in very warm conditions. We would have expected that all the samples should be more or less on the same spot, as they are all collected from Temple Lake, but this was not the case, as we have learned to our surprise. What we can say is that most of the samples, they are here more or less in the lower middle part of the diagram, here to see. And on this diagram, which shows the hydrogen isotope composition versus sulfur, we can also see it here. So we can assume that this one is the spot of Temple Lake, and it has an accumulation of the samples that have been collected there. Still, what we see is that we have feather samples that must have been formed at areas or localities which have more depleted water than the Temple Lake. And there are samples that must have been formed at localities which are very strongly, let's say, enriched in heavy isotopes with respect to the Temple Lake locality, which means that the assumption that all the feathers are produced during the molt at Temple Lake was incorrect. This is, on the one hand, was a bit irritating, but on the other hand, this was, let's say, very interesting and fascinating, and also a possibility to learn more about the migratory pathway of these geese. It showed us that they must have, let's say, that molting of the secondary feathers is not well defined, because it obviously takes place during summertime and during wintertime, and sometimes during different locations at the summertime and the wintertime. This can be shown by when we look at the hydrogen isotope values in combination with sulfur isotopes, for example, as we see it on the right side diagram, where you can see that we have some quite large variation. On the one hand, we can see here that we document the large variation in this, let's say, inner Central Asian or Siberian situation, where different water bodies seem to have very distinct and very different sulfur isotopic values. So we have quite low values here, we show it here, and quite high values here on the other hand. And we see that there's also some variation, but to a smaller degree on the right-hand side, where the feathers that have been formed during winter have been formed, so that we have a smaller variation in sulfur isotopes. If we look for other parameters, as for example for the carbon isotopic composition, we know that the feed is the relevant parameter. As the geese are, let's say, they are feeding on plant material, it is the plants they are feeding on. And so it can be expected that if we have high samples enriched in carbon isotopes, it must be the influence of C4 plants. A possible source of the C4 plants can be C4 grass. On this small map that is shown here on this slide, you can see the orange color indicating C4 plants domination globally. And you see this one hand is in India, and also in small dots in Central Asia, less well to be seen here right now. By nitrogen isotopes, the influence of C plants, for example, would be the reason, or let's say the consequence that we will have high 15N values, or if we are in very acidic areas, then this also might lead to, let's say, enriched delta 15N values. So if we look at these values, we can see here that we have, on the one hand, quite different values for carbon 13. On the one hand, we will see that we even on the summering side in Mongolia, that some of the individuals had access to a mixed C3, C4 plant diet. But we also see that in the wintering side, generally the carbon isotope values of these feathers have higher values than on the wintering, at most of the wintering samples from the wintering sites, which is in a good agreement, as we said that on the subtropical Indian peninsula, there is a dominance of C4 plants. For delta 15N, we see a smaller variation, and it's probably, let's say, of less relevance with respect to that. But we can see that in India, or let's say, let's be a bit more cautious and say, in the wintering side, we have a larger variation in 15N values. And let's say it might be different, let's say, reasons for this, either that they were feeding also on plants growing in fields, or it might be that they are feeding on plants in mangrove, deltaic, or even shallow marine water realm, because this might be the reason for high delta 15N values. So for the interpretation of data, as I said, it's very important to have necessary information, knowledge about the bird species, as is the molting patterns, migratory routes, times, stopovers, and of course the feed. And what we have seen is this, that up to now, so it seems, quite some of this data required is not available, because the assumed molting pattern is wrong, as we can more or less prove, so to speak. Still, this allows the interpretation of results with respect to the movement pattern. Some individuals have built secondaries further north at lakes with lower water isotopes. So it seems that even on the summering sites, there is some, let's say, short to medium migration during summering times. Then there are some secondaries that grew at stopovers or in transit from the summering sites to the wintering sites, because they have intermediate isotope values. And some of the secondaries grew at winter destinations. And it also showed that the birds individuals stayed at different wintering regions, because we also have quite a big variety. We could not have demonstrate an unequivocal influence of the sea. But what we also did see is that that we obtained 3 feathers of each individual cord, and we have very different, let's say, patterns, that sometimes all 3 feathers gave the same signal, or sometimes all 3 feathers gave a different signal each of this feather. So it's now difficult for us to estimate whether all the individuals have the same molting pattern, or some of the individuals have different molting patterns, which means that some shed all the secondaries at one spot and getting these homogeneous values for the 3 feathers, or that some individuals shed the secondaries throughout the year, and therefore you have very different isotope values. This is something that needs to be also still to be cleared, because it's not, we cannot answer this question so far. We have now made a short comparison or addition from these Mongolian samples with feather diet data from other countries. Here it's also from Albania and Bulgaria, where we can see it's also mainly feathers of waterfowl. For the Bulgaria, we see that we have there are also a few specimens which also have very low isotope, hydrogen isotope values, which is the x-axis, and therefore this is also waterfowl which stays in summer in let's say in Siberia and changes the feathers there. And for the Albanian samples, we see that there are two samples, two theta samples. These are the, let's say, the gray squares. And two of them have here rather enriched isotope values here for hydrogen. And these two are from a barn swallow, so it's not a waterfowl in this case. It's a non-waterfowl but migratory species, showing that at least these two feathers have been, let's say, produced during wintering time in most likely in Africa, in the southern realm. But still, we see that we have a quite similar range with respect to the feathers. Furthermore, what we can see is that if we compare the samples from Albania and Bulgaria, especially if we take into account the sulfur isotopes, which is here, here in the center, the diagram, we see here that we can differentiate the Bulgarian samples, which have lower sulfur isotope values, and the Albanians, no, sorry, it's quite the opposite. We have the Bulgarian, which have a very small range here in the middle, and the Albanian, which are having dominantly lower sulfur isotope values, which again shows us that there is a potential for stable isotopes to differentiate the geographic origin of biogenic materials. So just some general conclusions here. We can use stable isotopes to backtrace animal movements. Hydrogen oxygen isotope give information about the isotope ratio of the ambient water the animals live on. Water isotopes are usually geographically influenced, as I've said before, with respect to, let's say, the geographic position, distance from the sea, altitude, etc. And other element isotopes can give valuable additional information. However, the interpretation is not always straightforward. Very often it's not straightforward, as necessary data sets are not always available. For example, the global sulfur and nitrogen isotopes in soil and water, water isotopes in precipitation, even though for the latter there exists quite already a big database, or also the distribution of C3 and C4 plants, which is also not as detailed known as we would like it. Still, isotope analysis can help to discriminate different geographic origin. And with respect to waterfall, it is very important to know about the behavior, migratory routes, molting pattern, feed preference, etc., to be, let's say, able to make as many assumptions and interpretations as we want to do with the data. Specifically for the Mongolian samples, we can say that the assumed molting pattern is incorrect. Secondaries are built during any time of the year, so it seems that the migratory pattern can be verified based on existing knowledge of the bar-headed geese, as we have samples coming from southern realm. Some individuals molted at different sites differing from capture locality. And individuals stay over winter in different areas, so they don't all go to the same places in winter, even though they were all caught at the same place in summer. And in Mongolia or Central Asia, in the summering regions, there must exist water bodies having very different sulfur isotope values, as we have seen this in the feather samples. And with this, I am at the end of my presentation. I hope I have given you a quick overview of some possibilities of the use of stable isotope applications. But of course, this is just a very small part of application. Then with stable isotopes, you have the possibility to tackle many different questions. And it's just a matter of, let's say, thinking about what can be done or in which way stable isotope can serve you in unraveling some problems. Thank you for your attention.
Thank you very much, Dr. Horace. Thank you for sharing very interesting and fruitful presentations. I believe there would be new insight that everybody can get from this talk. And Mbak JG, could you present, could you stop the screen sharing? And Mbak JG will share some questions that you get. From your informations, now there are around 100 audience attending this expert talk. Is there an if there is a audience that would like to deliver the questions online, you may raise your hand, and I will give you a time, and I will call your name after I present the questions written in the chat box. So this is the first questions, Dr. Horacek from Nurfianto Giar Pangindu in Malang. The first question is, how is the application of stable isotope analysis in terms of country harvest origin standard and geological indications in plantation crop product like coffee, tea, etc., and fish product such as in Sidoarjo smoked milkfish. So Sidoarjo is a small area, regency in the Central Java. And the second one, what are the economic and financial prospects of stable isotope analysis in organic farming certifications? And the last one is, can stable isotope analysis also be used to identify the origin of non-timber products such as resin and essential oil? Could you directly answer the question?
Okay, I will try to answer this directly. For the first question, plantation crop products like coffee, tea, there is already now the beginning to control this by stable isotope analysis, as let's say, especially for products from geographically, let's say, far or let's say distant localities. It should be quite, let's say, quite easy and straightforward to differentiate them. For example, Indonesian coffee or tea and Chinese tea, or Indonesian coffee and let's say Ethiopian coffee. I would assume that it will be very, very rather straightforward and easy to differentiate these origins. But one has to check, but I would also assume that there is the possibility even to differentiate geographic origin within Indonesia. One example might be coffee from, let's say, Java or Papua or Sumatra, or there might also be the potential to differentiate different coffee farms on Java. But this is something that is not so straightforward, which will be more complicated because these localities on Java are geographically closer together. In this way, most likely representing ambient environmental conditions which are rather similar, and thus it will be more difficult to identify differences between individual coffee farms, for example, on Java. But it's something that should be tried, and there might be the possibility to differentiate too. For example, different altitudes, different geology, different part of let's say agricultural practice. So there is the potential also to differentiate these geographic localities. I have to admit, I don't know about this Sidoarjo smoked milkfish. If you mean that this type of fish is produced in Sidoarjo, if I'm not, yeah, but it might also be produced in other areas, then there also is the potential that you can differentiate, mainly due to differences in the water isotopes of the, I don't know, is it a lake or lakes or rivers these fish are reared in, or also the geology of the catchment area, or also that different feed is applied to this fish. But it's something that needs to be checked. And I have to admit, I really don't know anything about this Sidoarjo smoked milkfish. But nevertheless, I would say there certainly is the potential that this can be done. So what are the economic and financial prospects of stable isotope analysis in organic farming certification? Up to now, I would say stable isotopes are sometimes applied, but up to now it doesn't play a very big role, because usually to my knowledge, I'm not specialist in this, but to my knowledge, control of organic farming is done by visits of the respective farm and by checking all the let's say the data available of what they bought, what they sold, and things like this, what materials they used and applied. So I would say the application is rather limited because there is one problem with organic farming certification. Organic farming certification is a matter of definition. And there are different control bodies that sometimes or quite often having different definitions of what is organic farming. And so it might be also a problem to unequivocally differentiate between organic and non-organic farming, because in the, let's say, so to speak, worst case, I can have a farm which is producing completely as it is defined for organic farming, but it has not been certified for organic farming. And then it is it isn't an organic farming because it hasn't been certified for it. But there is no way to identify this by stable isotopes, whether this farm has been certified or non-certified, just on the isotopes. To your third question, of course, you can also use non-timber products such as resin and essential oils, but I would assume that in some cases, this resin and essential oils contain maybe less elements. Let's say that it might be that in this oil, there isn't a sufficient amount of sulfur to analyze it for its isotopic composition. And thus, in this way, it might be more difficult to differentiate between geographic origins. I have less parameters to investigate. But still, there is the possibility that you can do this. And if there is the need to differentiate between geographic origin of oil and or resin, you should try it. But it might be worth to combine stable isotopes with other analysis, like, for example, trace element analysis, strontium isotope analysis, and there should be further methods to add that you have more parameters for differentiation. I hope I've answered your question.
So, Mika, with regard to the second questions, is stable isotope can be used to differentiate whether the farming product is coming from the organic farming means the fertilizer uses organic fertilizer vs if the fertilizer use chemical fertilizers.
Generally, I would say yes, you can differentiate between organic fertilizer and conventional or mineral fertilizer, but there are some exceptions. For example, if you produce an organic fertilizer by, let's say, animal waste, and the animals fed entirely on leguminous plants, like for example, clover or beans, then this organic fertilizer might have a low nitrogen isotope value similar as the one of conventional fertilizer. And in that way, there are some limitations, as some fertilizers might not follow, so to speak, the general trend, because usually you say organic fertilizer has high nitrogen isotopic values because it's usually produced from animal waste, whereas conventional fertilizer usually has low nitrogen isotope values as they are normally produced from nitrogen from the atmosphere having low nitrogen isotope value. So it can be done, but there are some limitations.
Okay, thank you. The next questions from Willem Amoblegur. He asked about the effectivity of the use of stable isotope for the ecological landscape research. For example, in mangrove ecosystem, to measure the mangrove health status, and what kind of isotope you can recommend that you can give a recommendation for them if they want to use the stable isotope analysis as one of their measure for mangrove health status?
It's now a bit difficult for me to answer this question because I'm not sure whether I really understood the scientific problem there. With respect to health status, there might be the potential to use, for example, carbon isotopes, as usually when you have plants which are in a stress situation, usually incorporate more carbon isotopes than plants in a non-stressed situation, as for example, I've explained for existence of water stress versus the absence of water stress. But this, I don't know exactly about what kind of health problems these mangroves might experience. Thus, I have no bit difficulty to answer this question exactly because I don't know what are the influences of this, let's say, this non-healthy mangroves. What influences do they experience?
So there are several indicators for mangrove health status. We usually call it the mangrove health index to measure whether the mangrove ecosystem is still in a good ecosystem. It means can support the organism who live there, and the unhealthy mangrove means that if the ecosystem is already degraded. But the interesting point that I catch from these questions is whether we can identify or distinguish whether the mangrove that is live in the different ecosystem, such as in the near karst area or in different area in different island in Indonesia. Would that be possible to trace the origins of the mangrove?
I would say if you have an influence of karst, means you have influence of meteoric water there, and so it would be possible to identify such locations by either maybe hydrogen-oxygen isotopes to slightly differing isotopic value between seawater and meteoric water. Furthermore, also sulfur isotopes might help with respect to that, and also nitrogen isotopes. But with your first question concerning the health status, I'm still, let's say, not certain, but it might be most probably because I don't know enough about this mangrove research. Because when you say that the mangrove health status is deteriorating or deteriorated, I don't know right now what leads to the deterioration. What is the effect which deteriorates the mangroves? Is it, let's say, contamination of the water? Is it a loss of biodiversity? Is it, let's say, the cut-off of the mangroves from the open sea or whatever? I just don't know enough about this research, because you need to identify what are the reasons or what are the mechanisms leading to the deterioration of the mangroves, and then we can identify potential proxies to let's say, to identify the status of the mangroves.
Okay. Thank you very much for answering the question. We still have 30 minutes around 30 minutes, and we still have a lot of other questions. Can we move to the third questions? Okay, this is from Mr. Sutomo. The unique adaptation of C4 plant allowed them to endure arid and hot environment. They also experience isotopic depletion, so this can also lead to distinct isotopic signature with other types of plants. And what does distinctions this can be beneficial for?
Yes, you're right. The adaptation of C4 plants allows them to be less, let's say, to be more tolerant on let's say, arid conditions. Unfortunately, I don't know this isotropic depletion, what this means. So I cannot answer this kind of question.
Sutomo, do you still here? Would you like to have to ask directly? You can raise your hand so we can identify you. No? Okay. Okay, then maybe we still have time later on. So Dr. Horacek will stay in our laboratory, no, in our institutions until the end of August. So you are free to ask him everything and to have scheduled discussions. You can contact me, or you can also send email to you. Is it okay, Misa? Okay, thank you. And then we can move to the next questions from Mr. Adianto. How could the carbon isotope predict the age of wood fossil, and how old maximum of wood fossil can that can be predicted by the isotope carbon isotope?
The prediction of age of wood is done also with carbon isotopes, but not with stable isotopes, but let's say with decaying isotopes. So at least if we are dealing now with wood of a bigger age, so which means older than 100 to 150 years, then we can use carbon-13 method not to predict, but to analyze the age of wood and wood fossils. And I'm not a specialist in this type of analysis, but I know that the half-life of C14 is around 5,000 years or something like this. And I think more or less the limit of C14 dating is around a few 100,000 years, if I'm not mistaken. But this is, as I said, this is a completely different type of technique involved, and I'm no specialist in this, so I cannot give you much information on this, as it is C14 dating that is done for the age of wood fossils or also other biotic material. And how old maximum? Yeah, I would think it's a couple of 100,000 years. That you can date back these kind of materials, like by C14 isotope analysis. And
Yes. And the next
question
you can answer directly.
About how effective isotope tracing for cattle which are fed with artificial food, not feed naturally. Right now I'm not certain what artificial food is meaning. I mean, I know that there is some kind of so-called strengthening food, or where there's different types of let's say nutrients are put together to enhance the growth of the animals. But to my knowledge, this is just an addition to the food, to the feed, to the cattle feed, and I don't know about animals which are entirely fed on artificial food, because they need, to my knowledge, they need let's say a fiber-rich diet, which is the easiest way to feed them straw or grass or hay or something like this. If you feed an animal a completely artificial food, then of course you can more or less produce more or less the isotopic composition, or this isotopic pattern that you want to produce, because you can produce artificial material now with certain isotope values, which is done for scientific purposes, for example, to trace certain compounds through the metabolism or things like this, or this has been applied in fertilizing studies to learn where the fertilizer is moving in the plant, for example. And so this, of course, can be done if you are producing an entirely artificial feed for animals. But when we're now regarding cattle rearing, where you have some dominant cattle feed, let's say grass, straw, hay, or something similar, and some so I'll say some additions to this to this feed, then most likely, with some exception, of course, this won't influence the isotopic composition not too, let's say, too strongly. But of course, if you analyze, for example, strontium isotopes, which is a trace element, and you have this feed addition containing a high strontium concentration, then of course this can become a significant addition to the feed and thus also significantly influence the strontium isotopic composition of the kettle. But if this is not the case, then I would not expect a very strong influence of these food additions. I don't know if I've answered your question, but just let me know.
Okay, thank you very much, Misha. I always spell your name wrongly. So everybody, please remember that we are talking about stable isotope. It means not the carbon 14 who has the radio. It usually called as the radio isotope. So we move to the next question. We still have several questions. Thank you, Dr. Misha, for your very nice presentation. And is it true that the naturally occurring stable isotope of water and other substances could be used to trace the origin of natural resources? If so, can we also use this stable isotope to track carbon cycle in open water sea system like in seaweed ecosystem? Wow, you have very various questions. I think everybody is enlightening by your talks. Could you answer the questions?
Again, I hope I will answer it correctly, but I'm not sure whether I understood the question correctly. So for isotope hydrology, it is already used to investigate, for example, the catchment areas of springs, to learn the water coming out of the spring, where does it come from, how much retention time occurred before the water leaves the spring. And so this can be done and is done with stable isotopes, mainly by hydrogen oxygen isotopes. I notice is this mainly from the moderate climate region, because there I have this, let's say, the variation in the season, the annual season. So I have different precipitation isotope values in winter than in summer. And I can also mirror this, or I can investigate it is in the spring, and to see about the retention time, because the longer water remains in a water reservoir before it re-emerges from the spring, the less pronounced the seasoning is then later on to be seen in the spring. So I don't know what, if even I answered your question correctly, but the origin of the water can be traced by this. History, I don't know what you mean with history, because, well, history, I would say, if I really have old water, this is also is sometimes found in springs. It's either when the isotopic composition does not fit to the region, then it might be, for example, if it's old water, which stems from, let's say, from the Ice Age, thus it has a different isotopic value than you have in this area nowadays. But this you can either investigate by the oxygen and hydrogen isotopic composition, because it has a typical, let's say, depleted value, and you can do it also with C40, because then you have the age dating of the water, and you can also have an information of this. Sources, I think maybe this is the catchment area that you are talking about, and I think I have answered this question. Sinks is, yeah, this is also again the catchment area, if I understand you correctly. And interactions in water, it now depends on what you're meaning with interactions. Water has lots of interactions. It might biogenic interaction, it might be water-rock interaction. So I'm not sure what you are meaning right now, but of course, water-rock interaction might, there's the potential to be able to identify this by analyzing also the, let's say, the dissolved inorganic carbon in the water. but I'm not really sure what you are meaning with this. So if this is something relevant to you, please come back to me, or you can maybe ask a second question that I can better be able to answer this one. Carbon nitrogen cycles. This you can do in, let's say, in the ocean water, for example, with respect to global cycling. You can do this in lake water with respect to lake water environment. So this is done and can be done in different, let's say, realms or environments. And I think, yes, you can also use stable isotopes to track carbon cycle in open water sea system, like in seaweed ecosystem. You can use this, but of course, and I don't right now have the information on this. You need to know what is the question that you want to answer, what is what you want to investigate. So this is something that needs to be established first.
Okay, I hope that is answering part of your questions. Next, can we move to the next questions? So, other questions from Pak Agus Tampugolon, or any suggestion for Indonesia to achieve food security and nutrition in relation? Wow, this is far too far from our previous talk. I'm really sorry, Pak Agus. I think I should move to the next questions. Okay, from Pasuyadi. How is the application of stable isotope analysis to estimate sediment carbon source, the origin site of sediment carbon? So is it con ah oh, it's really difficult to spell. So you can read it. I think you can spell it better than me. So please answer the questions, Mika.
With respect to autochthonous or allochthonous, I would think if you are speaking about carbonate sediment, then this is easy because, or more or less, it can be, let's say, it can be easy because you just have to check the source of the carbonate, where it's coming from, whether this has the same carbon isotope values or different ones. If you're talking about organic carbon, then of course it's more complicated because it depends on from where the organic carbon is coming from. I don't know, perhaps this is related to carbohydrates. So let's say oil and gas in sedimentary rock. There I would say it's rather difficult to say whether it's allochthonous or autochthonous by stable isotopes. There I would rather say, is the potential there for the sediment to be a source rock? It means, does it from the beginning contain sufficiently carbon, or the organic carbon, that the oil and the gas can come from this rock? Or is the sediment rather poor in organic matter, and thus it's not likely that it's the source rock? And therefore, if we find there is some oil or gas in the cavities of these sediments, then it's rather allochthonous than autochthonous. But as I say, now I'm not really sure in which direction this question is going, and the accuracy as well. Please, here I would say the accuracy of measurements is one thing. For carbonate, it's less than 1 per mil. It's 0.1 or 0.2 per mil, the measurement itself. And for the organic carbon, it's similar like this. But if you're meaning the accuracy of to say whether it's allochthonous or autochthonous, here I would say I need to know more about the problem and the question that you want to answer before I can answer this question. Perhaps you can also come back to me with this, because right now it's not really clear to me what you're meaning. If it's with hydrocarbons, I hope I have answered the question. If it has to do with something else, please come back.
Yes, thank you very much, Mika. So Dr. Suyadi is work for the Center for Ecology and Technobiology, so you are very welcome to have more discussions with us here. And the next questions is coming from Ali. Can bioplastic autentifications use stable carbon or hydrogen isotope to distinguish between synthetic and bioplastics?
This is an interesting question. Yeah, I would say theoretically, I think it should be able, but it depends on the, let's say, the materials the bioplastic is made from. If this is, let's say, plant material, then I would expect that you should be able to differentiate, because the plant material usually has a different isotopic value than the one from oil derivatives. So, but if the bioplastic partially also consists of synthetic plastic from oil derivatives, then it will become more difficult, because you have a mixture of different materials that you're analyzing, and then might be more, let's say, a challenge to identify this. But if you are talking about, so to speak, pure bioplastic entirely made from, let's say, plant material versus a pure synthetic plastic, then I would expect that you have a high potential of differentiation between these two commodities.
Okay, we will move to the last questions. This is from Henny. She have several questions. The first question is, how effective is the use of light element isotope versus heavy element isotope? Second one is there any differences in C isotope composition among varieties within the species of C4 plants? Third question is, liquid can be analyzed using EA-IRNS using certain type of capsule and preparations. When or for what types of sample should we use LC-IRNS? What are the advantage and disadvantage between both? And lastly, will there be a shift in carbon isotope value over a long period of time? There are so many questions. It's very technical, but it's very important. I think she already conducted some research on the using the stable isotope and has several questions, technical questions too.
To question A, it depends on the question that you want to answer. As hopefully I've told you that the light element isotopes are mainly coming from, let's say, the water cycle, like hydrogen, oxygen, the fertilizer or the nitrogen cycle, the sulfur cycle, yes, there's or the carbon cycle, or let's say the feed. So it depends on which type of food you want to investigate. So it depends on which type of elements will be the most suitable ones. The heavy elements like strontium, or it might be lead, they are mainly geogenic parameters, or lead is also influenced by anthropogenic activity. So it depends on really on which questions do you want to investigate, how suitable are the different geographic localities that you want to differentiate. Do they have more or less the same geology? Then the geological parameters will not be very helpful. Do they have a very heterogeneous geology? Then it might also be problematic, because it will have in this area a wide range in the heavy element isotopes. So it really depends on the question and on the exact situation that you want to investigate to determine which parameters are more effective than the others. Is there any difference in C isotope composition among varieties within the same species of C4 plants? Yes, there will be probably due to differences in the geographic origin. You will have differences in the same species of C4 blends if they're coming from different. You might have differences if they come from in the C isotope composition, if they come from different localities. I'm just thinking now whether I have a good example. Right now, I only find examples from, I know from CAM plants, because vanilla plant is a CAM plant, and you can differentiate them also by differing carbon isotopes. And the same also is the same for ananas. But for C4 plants, let me think, I have to check about sugarcane if I have if I have analyzed sufficient numbers of them. But most probably also you will also have some differences in for the same species in C4 plants depending on the geographic origin, the locality. Liquids can be analyzed using IRMS, yes. This cannot be done by using this type of capsules, but you can also mount a liquid injector on the elemental analyzer. And in this way, you don't need to put them in special capsules, but you can just inject them with a syringe through a septum into the elemental analyzer. This is used if you want to analyze the entire liquid, like for example, oil or water or alcohol, then you can use this either capsules or liquid injector to analyze this on an elemental analyzer connected to an IRMS. When you want to use LCM IRMS, you want to separate by liquid chromatography the different compounds of a liquid and subsequently analyze these different compounds. For example, if you have a sugar solution, you can inject this, or you can put it in a liquid LC-IRMS. The liquid chromatography separates the different sugars you have in this solution, and then one after another, these sugars are coming in an IRMS and are separately, but during within one run from one sample, one solution, one sugar compound of the other is investigated, for example, for the isotopic composition. So you see, there are different aims that you want to achieve. There are different goals that you want to have for using these different techniques. Because for IRMS, if you inject the sugar solution, you get the bulk, you get the average isotopic composition of the entire sample. For LCMS, the different compounds are separated and are measured consecutively one after another. And will there be a shift in carbon isotope values over a long period of time? This is now, again, a question which is now difficult or easy or difficult for me to answer because it's very general. If you mean, if I have a sample material and store it over a long time, will there be a shift in carbon isotope values? No, there won't be any shift in carbon isotope values as long as this material is not changed. For example, if I have a piece of meat or if I have some oil and I deep freeze this oil or I deep freeze this meat and no biogenic activity or microbial activity is taking place in this sample, then the carbon isotopic value will not change. As I said, we are talking about stable isotopes. Stable isotopes remain stable as long as the material remains unchanged. If you're talking about carbon isotope values over a long period of time with respect to, I have analyzed grass samples which have been harvested in 1960, and I'm going to analyze from the same spot grown under the same conditions grass samples harvested this year, then I will have a shift in carbon isotope values, even though it's exactly the same condition, it's exactly the same spot, but it will have a slightly different carbon isotope value because the CO2, the isotopic composition of the CO2 in atmosphere has changed over this period due to anthropogenic activity. So it now depends on what you're meaning with the shift in carbon isotope values over a long period of time. If it's a material remains inert, then there is no shift. But if let's say the environmental conditions are changing, then the carbon isotope value also might change.
Okay, thank you very much for answering all the questions. To you all, Dr. Horacek will stay in Cibinong, Indonesia until the end of next week. We mostly will be in genomic laboratory, and you are very welcome to contact and discuss more after today. For your information, Berlin has two isotope review mass spectrometry or IRMS, one in sample and other one in genomic laboratory in Cibinong. So we can use this machine to conduct the research related with stable isotope. Thank you very much to Dr. Misha Orasek and also all audience who already spent their time to listen to this expert talk. I really hope that this talk will give you all new insight and inspiration for future research and collaborations. Lastly, and thank you very much for listening to my talk. And lastly, I will give back this forum to Pak Bugi. Maybe you have several announcements. You will get the door prize for the questions. Thank you very much, everybody. Bye-bye.
Bye. Thank you very much, Bu Ruli. It's very interesting discussion, both Pak Misha and moderated by Bu Ruli. But actually, this is for me is beyond my topic. But very interesting listening to Pak Misha presentation and how Pak Misha deal with the answering the questions. Thank you very much, Pak Misha. And I think that some points I will try to inform to the audiences. The first is about the door prize. The secondly is about the continuing of Buriana information that Pak Misha will still here in Bogor or Cibinong office until the end of August. So if you have any further questions for discussion, you can contact Buruli to make an appointment to have a discussion with Pak Misha. The first one is about the third prize. So Mbak JK, maybe you can provide the slides who will win the door prize. Okay, so we have already decided that based on the Q&A sessions, we have chosen the best questions. First is for Bu Nurfianto, Pak atau Bu Nurfianto Giar Pangidung di Malang. And then Ibu Heni dari TPR. Okay, so Mbak JK will later on contact these two winners. And also we are going to inform also that the list of attendance has already shared in the chat box. So it will be provided until 5 p.m. this afternoon. And Pak Kapus is still here to close the expert talk. If not, maybe I will give it back to Buruli to close the expert talk for closing.
Well,
With some insight, I think so you can attract audiences to contact you or to have a discussion with Pak Michelle later on. Please, Buruli. Thank you.
Thank you very much, Pak Bugi. It's such a sudden request. First of all, thank you very much for listening to this expert talk. I think this kind of expert talk is very important for us as a researcher to share some new knowledge, new informations, new innovation that can be very important to build a future research and future collaborations. I think Dr. Horacek is very open to discuss and to have more collaboration or to interact with you all in the future. And for future discussion, you can first contact me. I put my email address in the chat box. You can send email, and then I will communicate it with Dr. Horacek to schedule a good time to have a discussions. We will have most of our time to conducting some experiments in genomic laboratory. So we need to schedule for a discussion. Again, thank you very much. I hope that we can have this kind of talk with another expert that can share their experiences, their new research and innovations uh, for us, so that um, we can have more information. It's difficult to close something. So again, thank you very much for all. And I think I wouldn't speak so much, too much about this, Fabugi. And have a nice evening for everybody in the Eastern Indonesia and for you all. Thank you very much.
Thank you very much, Bu Ruli, and please give a big applause to Pak Misha and Bu Ruli for having this second Expert Talk very smoothly and running well. And for all audiences, see you again on the third episode of Expert Talk in the next 4 months. Thank you, everyone. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh. Bye. Pak JB, I'll leave it to you.